Hello, guys. 12th graders. Let's do some practice questions on the first unit. I have prepared about 20 questions. This may help you for your monthly test coming soon next week. Therefore, let's do the questions together. Okay. The first question: when a biotechnology industry is said to be environmentally compatible, it means all except, of course, environmentally compatible. We are talking about the environmental aspect. So it should be eco-friendly with the environment, of course. Just B, prevent loss of biodiversity, exactly. C, reduce or prevent pollution, exactly. D, social equity. Of course, this is the social aspect. You remember, three factors must be considered as far as we're talking about biotechnology utilization. One is the society. We have to consider the society. Two, the environment. Three, the economy. This should be integrated whenever we are talking about sustainable development. Therefore, here it talks about biotechnology industry that considered environmentally compatible. When we say it talks about the environmental aspect, therefore T is the answer, which is not included here. The second question: which, if any, knowledge in biology may not be utilized directly in biotechnology from this given? Genetics, perfectly. We do utilize genetics, specifically genetics at molecular level. Transferring genes from one organism to another is highly utilized in biotechnology. So that we can produce transgenic organisms, or even we can apply it into the medical field of the technology to cure genetic disorders. Genetics is highly utilized, of course. Microbiology, very important, very important. Starting from isolating microorganisms to characterization, screening out the best for particular purpose. Microbiology is highly utilized. So we do require in biotechnology, we do require specialists in microbiology, specialists in the knowledge of genetics. Then molecular biology, no different from the genetic engineering, of course. And cell culture technique, exactly. How can you grow without the knowledge of cell culture technique? How can you grow and characterize, screen out microbes that have been isolated from certain habitat? Impossible. Therefore, the answer is none. All these specific fields are required in the biotechnology world. Third, which recent development in biotechnology directly works in repairing or replacing damaged tissues or organs through techniques such as tissue engineering, stem cell therapy, and gene editing? Of course, self-answering question, of course. Which one? The first, biofarming. Biofarming means when we are genetically engineering plants so that they may have some special traits like large fruits, or they may produce, even we can insert genes into plants so that they can produce vaccines, chemicals that are used as vaccines. Even it's possible to make plants produce milk by genetically engineering milk producing genes from mammals into plants. Therefore, biofarming includes all those. The second, regenerative medicine. Transgenics is transfer of genes, not, of course. Here the question answers the recently developed biotechnology technique that is involved in tissue engineering. We engineer tissues, stem cell therapy, stem cell, special groups of cells that can multiply, differentiate, and give rise to organs or the whole organism if it is embryonic stem cell, of course. Gene editing, all these are under regenerative medicine, of course. Regenerative medicine. Transgenics talks about transferring genes. The cloning is, of course, taking a single cell from animals and trying to work on the genes so that the cell divides, differentiates to give rise the whole organism. The other version of tissue culture. That means plant tissue culture can give you the whole organism. If you take now a recent development in biotechnology, of course, cloning. Even they have cloned mammals like sheep and cows and other mammals, and they are telling us it's possible even to clone humans. Of course, there are many ethical questions behind it. It's not allowed. The technology not allowed to clone on humans. Cloning humans is totally erased from the sphere of science. Otherwise, scientists are telling that it's possible to take one single cell, just single cell from humans, and work on the genes so that the cell is provided with nutrition and suitable conditions so that it divides and differentiates and to give rise to the organism from which the human being from which the cell has been taken. If you take one cell from the Malacca and clone it, it will give you exact copy of the Malacca. That means, therefore, sexual reproduction in humans artificially brought about. Therefore, it talks another topic, transgenic, another biofarming, another. Therefore, the answer here is regenerative medicine, of course. For the SCP, single cell protein, now by the name protein. Remember, protein is protein extract from this bacteria. That is C. Methyloficus methylotrophus. This is the bacteria that has been used to produce proteins at industrial scale, of course, to be used as feed for animals. And 5, weight contains this protein. Weight contains this protein, which makes up more than 80% of the protein in weight. It is responsible for deformation and also gives elasticity to the death. The answer is very clear. That is gluten. 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 And gases such as CO2 released during microbial respiration produces porous and spongy texture in the bread during baking. And this is, of course, you know it. The answer is D. Leavening. Make it rising of the dough so that when you bake the bread is spongy, fluffy, with many small pockets of air. Mycoprotein is mainly produced from this genus of fungus. Of course, it is Fusarium, specifically Fusarium venenarum. Venenatum. This is the main gas responsible for spongy texture in bread or injera, and also imparts this flavor. Of course, the gas is CO2, not only CO2, but the most dominant gas produced by respiration. Aerobic respiration or anaerobic respiration produced by yeast produces CO2. Yeah, CO2, and CO2 imparts CO2 produces some sort of carbonic acid with water, reacting with water in the bread, so that it gives tangy flavor to the bread. The answer is CO2 tangy D. Number 9. What is exploring molecular, genetic, and species level diversity for products of economic importance called? What do we call this? When we explore living organisms at molecular, genetic, and species level, their diversity at these 3 levels for products of economic importance to exploit their products for economic importance. What do we call it? This process in biotechnology is known as bioprospecting. Simply bioprospecting. You can see, no need of knowing it. Just if you are simply introduced about biotechnology, you can. Biopiracy is doing harm by using biotechnology. It's part of dark biotechnology. Of course, biofuel is production of fuel from biological sources, and biodiversity talks about the diversity of living organisms. Therefore, here we are talking about molecular, genetic, species level exploring in order to extract chemicals that have economic importance for prospecting. Number 10. These are all the qualities of the protein-rich food sold in England by the name corn. Protein-rich food that is obtained from fungi, that is Fusarium. What do you call it? Mycoprotein. Then these are all qualities. Therefore, this food high protein content. Exactly. That's why we call it mycoprotein, protein-rich. Having chicken flavor. Yep, I told you this in the class. Very delicious chicken flavor, and enough fiber. Yes, it has enough fiber for your faces to be eliminated easily. No need of will not develop constipation, of course. And no fat content, no, this is wrong. It has a very small amount of fat, very small amount of fat. We can't deny it. Just it doesn't mean non-existent. No fat content is wrong. Very small amount of fat is a right description. One of the following differs from the rest: brandy, champagne, gin, and cognac. How can you differentiate this? Remember, fermentation only alcoholic beverage and the spirits. One of this is fermentation only product, of course, that is a bubbling wine. They call it bubbling wine because it is packed with carbon dioxide. When you shake it and open it, it bubbles. That's champagne fermentation only, alcoholic beverage. Otherwise, brandy, gin, and cognac are what? They are fermentation plus distillation, hard alcohols, alcohol content 40 to 60%. Which phenomenon explains that when a species becomes extinct, the plant or animal species also become extinct that are obligatorily associated with that host species. When a species becomes extinct, the plant or animal that is associated with this extinct species will slowly become extinct. Which terminology describes this, of course? Aquaculture? No. Fragmentation? No. Invasion? No. Co-extinction. They become extinct together. Therefore, co-extinction is the terminology appropriate here. And 13, when potato is for vodka, dash is for brandy. Brandy is made from, remember, brandy is fermentation plus distillation. And what is fermented first? It's grape juice. If you like further information, of course, I have here next slide. Look, whiskey, especially the Scottish whiskey, is from barley. Scotland. The people of Scotland produce their delicious whiskey, most like whiskey, by first fermenting barley. Other countries use wheat, corn, rye. All of these are used in other parts of the world. Otherwise, when we talk about whiskey, Scottish whiskey, it's barley. Now, bourbon, this is another hard alcohol, bourbon, fermentation of corn or rye, bourbon. Brandy and cognac, they are from wine or grape juice. You ferment grape juice, it produces wine, then you distill the wine to produce the brandy, cognac, the hard alcohol. Vodka is for potato. Remember, Russians love vodka. Rum is from molasses. Molasses is um, byproduct in sugar refineries. Of course, it is full of sugar. Therefore, that sugar is used for fermentation. Then distillation. Then it gives you this hard alcohol known as rum. Gin from barley, of course, just like the Scottish whiskey. There is Fenny type of, especially now in Asia, Fenny apple juice made from apple juice. These are for your information and don't forget. Therefore, here it says, here, this bacterium has been utilized in agricultural biotechnology for transferring desired genes into plant cells. Of course, you will learn about this. Of course, not yet. Talking about genetical engineering plants specifically. This bacterium, which is the natural genetic engineer, this bacterium, when it infects plants in between the stem and the root junction, it infects plants and it inserts its genes carried on plasmids. Genetically engineering bacteria. Scientists learned this and they are using now agrobacterium tumescens to transfer the gene that they need into plants. Therefore, it is one raw material for plant geneticists to transfer genes into plants. Therefore, the answer is C. Which one of the following is said to be the most important cause or reason for the extinction of animals and plant? Of course, this is clear. This is clear. It's the first one, loss of habitat and fragmentation. Loss of habitat. Humanity is expanding, using forests, forest clearing, destroying forests for agriculture or industrialization. This is the most destructive part, which is causing huge extinction, especially of the equatorial rainforest areas. What are the excess? What are the excess and the unsustainable use of resource called? This is overexploitation. The strategies and actions that have the objective of meeting the needs and aspirations of the present without compromising the ability to meet those of the future. Yes, that is sustainable development SD. Remember that. Conservation strategies particularly are focused on conservation. Primarily, conservation strategies are focused on biodiversity, diversity, genetic diversity, species diversity, molecular. This is studied under the biotic resources. We have to focus. When we focus on the living resources, plants, animals, microbes, we can also care about the non-living. Therefore, primarily the biotic resource. A is the answer. The last question. Pick the odd one out. It says vitamins, antibodies, antibiotics, and alcohols. Give you 5 seconds, please. No answer? I leave this for you. Answer this. Thank you very much. Thank you very much. Work on these questions, and next week you will have one nice test. Thank you.